Hi, thanks for being here today. I'm really excited to talk about my group's work on patterns of growth and thickness in the cerebral cortex. So first of all, I want to talk about brain morphology. The brain during gestation is changing both in size and shape. So it's getting, as you can see from these images here, not only much larger, but also developing the characteristic wrinkles and folds that human brains are known for. And so as this morphology is taking place, the structure of the brain is developing, and that of course is related to the function. The particular aspect of brain structure that my group is currently most interested in is cortical thickness. Cortical thickness, which is just the thickness of the outer layer of the brain called the cortex, is an important biomarker of neurological health. So abnormalities in cortical thickness have been known to be associated with a number of different mental and neurological disorders, including autism spectrum disorder, schizophrenia, and epilepsy. I want to be clear here that when I talk about cortical thickness, I'm not interested in only a single scalar number that's the average cortical thickness throughout a brain. There's a lot of research that's been done in this way, and it can be very, very helpful to look at averages on that scale. But the downside of that is that really a lot of information is lost in the averaging process. So I want to illustrate what I mean by that. This is the typical human brain divided into about 70 regions per hemisphere, and each region is colored according to its average thickness. And so you can see that there is quite a range of cortical thickness throughout the brain. And of course, if we were to average this across the whole brain, then we would reduce all of this to a single number and capture no variation. In addition to this, we can look at it in an artificially inflated view of the brain. And this will make visible some of the surface area that's buried in those folds that we otherwise can't see in the peel version above here. So what's happening here is that when the brain folds, we see gyri, which are the sort of mountain peaks of the brain, and sulci, which are the valleys of the brain. They form. And in the typical peel view, what you would see if you looked at an actual brain, you're really only seeing the gyri. And so in order to see the sulci, we have to artificially inflate the brain in some way. So that's what I'm doing here. So this is great. This is a regional approach, averaging cortical thickness across a small area. And while that has its value and really can capture a lot of variation that we do see in cortical thickness, we can also look at a local level. This would be a vertex-wise level where we have around 70,000 vertices in each hemisphere. And we can look at the cortical thickness at each of those hemispheres. And if we do, we see something like this. So again, you can see this broad range of cortical thickness throughout the brain. You'll notice that it's varying on a much smaller scale than these regions that have been defined on the left. And again, you can see that if we artificially inflate the brain, we can see really the entire surface area. So we have chosen to use this local approach, this vertex-wise approach, to really capture the fullness of the complexity of the patterns of cortical thickness throughout the brain. So the overwhelming trend in these cortical thickness patterns is that sulci tend to be thin and gyri tend to be thick. So this has been known for a long, long time. You can actually see it just with your eyeballs looking at these MRI images. But in a paper towards the end of my PhD, we analyzed cortical thickness data from around 500 individuals and showed that this difference is statistically significant, that gyri overwhelmingly tend to be thicker than sulci. And so the question that arises out of this is why does this happen and how does it happen? And it's of course easy in any biological system to come up with a biological hypothesis. So the way that the cortex forms is that these intermediate progenitor cells migrate along radial glial cells and become neurons and take up residence in the cortical plate. So it's easy to imagine that if you just have more of these neurons migrating towards one location, and then that location is designated to become a gyrus, that you would end up with a thicker gyrus and conversely a thinner sulcus. So that's of course possible, but towards the end of my PhD, I realized that actually I was seeing these similar patterns in our simulations that had no such source of biological heterogeneity. And so that led me to develop a mechanical hypothesis for these cortical thickness patterns. And in a paper that was published in 2018 with my PhD advisor, Ellen Kuhl, and some collaborators, we investigated this more fully. So here I'm showing results of a computational simulation where we started with a uniformly thick cortex, had it grow homogeneously throughout, and saw what happened as it began to buckle and fold. So here I'm reporting the gyral sulcal thickness ratio, and what we can see is that shortly after the buckling point, the thickness ratio is very close to 1, or basically that the gyri and sulci are similar thicknesses. But as growth continues to happen, we see that the gyral sulcal thickness ratio increases pretty significantly to this point where the gyri are almost twice as thick as sulci. We did similarly a physical analog experiment using stretched polymers, and again found a similar thing, that we can see very high ratios of gyri to sulcal thicknesses in these simple physical models that have no biological source of heterogeneity. Later on, we were able to show that this is actually a phenomenon that's unique to soft systems. So when I say soft, I mean that we often think of these materials as a bilayer. So we have in the brain a subcortical substrate with a thin film representing the cortex on top. And if these materials have similar stiffnesses, and in particular, if they're both soft, then that would be a soft system. So here I'm tracking the thickness ratio over the time of our simulations. And what we see is if we have a stiff film on a soft substrate, or if the cortex happened to be very thick, then we don't really see the development of a very large thickness ratio. Really, the two thicknesses in gyri and sulci tend to be very, very close. However, if we have a soft film, which is actually the behavior that we see in the cortex, then you see around the point of buckling this drastic increase in this thickness ratio, and it remains high. We can also look at this at any given time point and look at it as a function of sort of versus the stiffness ratio. And what we see is that when the stiffness ratio is large or close, gets close to 10, so the film is around 10 times stiffer than the substrate, then we, again, don't see a significant thickness difference develop. But for lower values, when the two materials have similar stiffnesses, then we do see the emergence of this really large thickness ratio. And again, that's the regime where the brain is, and really other biological materials often. So all of this work has led us to consider, of course, this mechanical hypothesis, but it doesn't really capture everything that we see in the brain. There's a small quantitative difference between the patterns that we see in our mechanical only folding simulations and data in human brains. So we decided to develop a biomechanical hypothesis that includes aspects of both of these and see how close that can get us to patterns that we see in human brains. So this is work that was done by my postdoc, Shuilin Wang, and the question that I posed to him is, how can we model heterogeneous growth in gyri and sulci? And he came up with a new computational model where the volume growth in the cortex is dependent on the mean curvature. So let me go through this equation really quick. Theta G is the volume ratio due to growth, so how much the volume has changed because of growth happening there. And this equation can change both temporally and spatially. And so here, this equation that I'm showing is an equation for the growth rate at a given time and point. G cortex is the baseline cortical growth rate. So in the absence of other things, that's how fast the cortex is going to grow. Kappa star is the mean curvature normalized by the cortical thickness. And so this is used to give us some indication of whether a point is located in a gyrus or a sulcus. So what that looks like is if this is our simulation and we've seen buckling start to happen, in gyri, the mean curvature is known to be negative, and in sulci, the mean curvature will be positive. And so again, that gives us a rough idea of whether a point is located in a gyrus or a sulcus. So I want to be clear here, we're not claiming that cells or tissues sense the mean curvature around them and respond to that actual curvature, but rather that they're likely responding to something, whether that's a chemical or a mechanical or biological signal that's cueing them to have different behavior in these different regions. And then finally, we have our curvature, which is a parameter that allows us to adjust the growth heterogeneity. So what that looks like is if we set our curvature equal to 0, we will recapture the behavior of homogeneous or uniform cortical growth, where this growth is not dependent on mean curvature. However, if we set our curvature to a negative number, then we will get increased growth in sulci and decreased growth in gyri. And if we set that number to be greater than 0, then we would get increased growth in gyri and decreased growth in sulci. And so this allows us to capture these 3 separate cases of homogeneous growth, preferential sulcal growth, and preferential gyro growth. So now I want to show a couple of representative solutions. This is for the case of homogeneous growth, where volume change due to growth is coded by color. So you can see that the growth field here is indeed homogeneous. And if we look at the case of preferential sulcal growth, then you'll see that there are regions of increased growth buried here in these sulci. And for preferential gyral growth, you can see that the visible tops of the gyri are growing faster than the buried sulci. So this just demonstrates that we are sort of achieving what we want to achieve with this growth model. And to assess the validity of these different cases, we compared the thickness ratios that resulted to data from around 28 humans. And so the human data is shown here in green with decreasing confidence intervals and increasingly light shades of green. And then the data points from our simulations are shown in these different colors. So what we see is that sulcal growth and homogeneous growth just aren't really capturing the data that we see in human brains. The thickness ratio is even higher than that. And instead, this human data really coincides with our cases of gyral growth. So what that has led us to conclude is that gyri are preferentially growing in some way, whether that's due to cell migration or increased cell proliferation in the cortex, or perhaps something like dendritic arborization, that is happening preferentially in gyri to further increase the thickness of gyri and therefore further divide the thicknesses of gyri and sulci. So that work was published a couple of years ago. And since then, we've been starting to think about tangential folds. And what I mean by tangential folds are sort of this folding in the plane that we see in the brain. To again use the analogy of mountain peaks and valleys for gyri and sulci, if you think about a mountain peak or a set of peaks, they don't always run in a straight line. For instance, if you're looking at them on a map, they might instead bend and curve and swerve. And gyri and sulci do the exact same thing, as highlighted by several of these lines on this brain. So the challenge of thinking about tangential folds is we're now thinking about the full 3D folding shape of the brain and not simply 2D gyri and sulci sort of curves and maybe very simple simulations or brain slices. So there are regions throughout the brain that are not only concave or convex, which we might sort of very naively assign to be sulci or gyri, but instead we have locations that are really a combination of both, that are concave in one plane, convex in another plane. And we call those saddle shapes, of course. So to capture this complexity, the complexity of these 3D shapes, my graduate student, Nagihan Demirci, whose work a lot of this is, found out about the shape index, which is a single scalar number that captures 9 different shapes here as shown. And so I want to emphasize that our approach is using topology, objective, quantifiable measures of curvature, and it is not necessarily anatomy. So anatomy would be some distinction classification made by probably a trained neuroanatomist who would draw dividing lines between this gyrus and that sulcus and things like that. So what I'm showing here in this image is the peel surface of the brain, and you can see very visibly a gyrus snaking through here. But within this gyrus, we have a number of different topological shapes. So we have some locations that are a typical convex gyrus, just like sort of the classic gyrus that you would think of would be. But also within this, we can find a saddle-shaped gyral bend, where a gyrus is bending really sharply on itself, creating a saddle-shaped region. And we can also find local concavities found on a gyral peak. And that, again, is going to highlight the difference between topology and anatomy. So we're not trying to make anatomical claims about the divisions that we've created in the brain. We're thinking about the topology and the curvature. And what this allows us to do is to really get on a fine spatial scale the information about the topology, about the curvature, which is going to tell us something about the mechanical state of the cortex in that area. So again, this is work done by my graduate student, Nagia Handamarchi, and what she was able to show is that this full three-dimensional folding actually does affect the cortical thickness. So here's a brain with its cortex colored by the shape index in that location, again, with that color bar shown here. And what she found is that if she divides points into every point falls into one of these 9 different shapes, and we look at each of those shapes separately and the distribution of cortical thickness of those points, we see that the most concave points tend to be the thinnest, the most convex points, here in dark blue, tend to be the thickest, and the other points in between fall in between in a smooth spectrum. And so what this shows us is that this concept of a binary division between gyri and sulci is perhaps not that useful, and instead we need to be thinking about the full three-dimensional folding patterns seen in the brain and how all of those aspects of curvature are really affecting cortical thickness. So this paper was recently published in Human Brain Mapping, and now I want to show a little bit of preliminary data on some things that we've been working on since then. So she's also been looking at folding patterns and thickness patterns in non-human primates. So these are, here, human is on the left, and then we also have chimpanzees and macaque brains. And we see, again, very similar trends of cortical thickness in them, with the most concave points being the thinnest, the most convex points being the thickest, and the points in between falling in between in thickness as well. So these 3 species we had a large number of specimens for, but we've also been looking at other primate brains where we have only a single specimen, but I think we can still really draw some interesting information from those. So here, just to fit a lot of information into one graph, we've collapsed the 3 most concave shapes into a single yellow color, the 3 saddle shapes in the middle into a green color, and the 3 most convex shapes into a single blue color. And we've arranged the brains here in order from smallest to largest based on their surface area. Although note that the x-axis is not to scale, it's ordered relatively, but there's not a uniform gap between all of the surface areas. So these are the 3 smallest monkeys that we've looked at. And what you can see, first of all, right off the bat, is that there's a general trend of increasing cortical thickness with brain size. We'll see that throughout the rest of this data, and this is a known phenomenon or scaling feature. But the other thing I want to highlight is that the difference between these thicknesses is smaller in the smallest brains and gets larger as we go on. We see that as well in this group of medium-sized brains. There are some outliers. Perhaps this rhesus macaque is thicker than than its nearby primate neighbors. But still, we see, again, this general trend of increasing cortical thickness, which again is a known scaling feature. But we also see this increase in thickness differences. Similarly with the large brains, the chimpanzee data that I'd already shown, but as well a bonobo and a gorilla, and then with humans. So again, the 2 patterns we see here are increasing cortical thickness, but also increasing cortical thickness differences with larger and larger discrepancies between these concave and convex thicknesses, or you might think of them as being similar to gyral and sulcal thicknesses. So to wrap up now, we've concluded that, first of all, cortical thickness varies really dramatically across the surface of the brain. So we really need to be thinking about cortical thickness patterns and how it varies across the brain. But then when we think about these patterns, we know that they're a product of, first of all, folding and soft systems. So we'll only see this in these materials that have similar stiffnesses, like the brain. We believe that they're also a product of increased growth in gyri, where gyri grow preferentially to sort of further exacerbate this thickness difference between gyri and sulci. And then also that the full three-dimensional folding patterns of the brain will affect these cortical thickness patterns. So moving forward in the future, we're interested in investigating these patterns further and understanding how they are similar or different in these different cases. So first of all, throughout development, during gestation as the brain is developing, what is happening to these patterns? Do they change and how? Also throughout phylogeny, so I just showed some preliminary data in a few non-human species, but there are a lot of species out there that we'd be really interested in looking at. And also throughout health and disease, what do these patterns look like in different neurological diseases and disorders? How could that information help us with diagnosis or potentially with treatment? I'm really happy to announce that our work on these first two bullet points on especially development and phylogeny has been supported very recently by an NSF Career Award. So I'm really looking forward to what the next 5 years holds for us and the things that we're going to find when looking at human development and mammalian evolution. Finally, I want to acknowledge the work of my students and my research group, particularly Shuolun Wang and Nagyahan Demirci, who did most of the work that I presented today. Thank you also to our funding sources. And if you're interested in reading more about the work that I described today, these are the two main papers I discussed. You can check out our lab website as well, and I'd be happy to take any questions now or via email. Thank you.